in a state called metaplasia or dysplasia, and eventually become the invasive cancer cells. And previous research has shown that gastric pits is one of the major origins for the gastric tumors. And therefore, for the first agenda in our talk, we want to find the gene expression changes between the cell stages that I have mentioned about. And we have a very nice data set, which is the next slide. So in our data set, we have different time points or cell conditions in the stomach. Here we have normal cells. We have the intermediate inflammatory cells. Then we have the metaplasia state where the cells are losing their functions, and eventually the cancer cells. So the first thing we did was to perform pairwise differential expressions across these conditions. And because we have a lot of comparisons, here is only one of those examples. So on the left panel, you can see that this is the comparison between the normal and cancer cell. And down there, we have the genes that are statistically significant. And the most significant ones have the ADR of 0. We have highlighted them and their names on the figure on the right. So if you see the color, these genes, and we have investigated their functions, they belong to the secretory cells in our gastric pits. And the other thing is that they are upregulated in our normal sample cells when compared to the tumor cells. And this observation is actually consistent across other comparison or DE tests. So in summary, we have taken these genes out, the genes that belong to the secretory cells, and summarized them on a bubble plot. So you have the y-axis, which are the secretory genes and that are statistically significant in our DE test. And we had different conditions such as normal and inflammatory, cancer, and metaplasia. So what we see is that these genes are more expressed in normal and inflammatory state where the cells still have their functions, whereas during metaplasia and cancer, these genes are downregulated. And actually, this observation has been validated using another gastric cancer data set. So this data set also have the normal and gastric cancer cells. And using the same genes, we see that they are more expressed in the normal cells, which poses a question or a hypothesis. We believe that the secretory cells have somehow been altered or even gone in the cancer cells, and therefore we went on to investigate the cell composition in our data set. So we have a feature on bBrowser X that allows you to do so, and this is the result that we found. Next. So again, we have 4 different cell stages, right? Denoted by 4 bars right here. And we had different cell types. And from the normal cells to the inflammatory state, we have a very big blue bar that represent the pit mucus cells. However, when we get to the metaplasia state, this population of cells shrinks and they no longer exist in cancer cell. And likewise, we also observe that the neck-like mucus cell in the normal sample has disappeared over time. And the second observation is that in the cancer population, we have now more metaplastic stem-like cell, which is the yellow bar here. So our hypothesis is that perhaps the mucus cell population has transformed into the cancer stem cells. And now we only focus on the mucus cells and the cancer stem cells and try to see what they have to do with each other, or how do the mucus cell transform into cancer stem cell. So now we do a step of subclustering and to perform an analysis, which is called the pseudo-time analysis. And I believe Julie can help me with this process.
Yeah, and so this is where I can demonstrate how to use pseudotime analysis on bBrowser X Pro. So the aim of pseudotime analysis or trajectory analysis is to see how cells change from one state to another. And we want to see what kind of genes are contributing to that transition. So firstly, what I want to do is, since we're only going to be looking at our mucus cells and our metaplastic stem-like cells, I'm going to subcluster those first. Subcluster the mucus cells and this. and I'll click subcluster. Okay, and from here I can perform pseudotime. So pseudotime is pretty simple. You just have to input the 2D embedding, which is the visualization that you want to view. I'll leave it at that one and click run. And these are the results from pseudotime. Okay, so from here, now we want to look at the metadata because we don't know how our cells are colored. So I'll choose the cell type metadata to see how our mucus cells, which is the light blue here, transition into the metaplastic stem-like cells, which is the yellow-green here. So typically, I want to choose the start of the trajectory around this area. It might be hard to see when I'm showing my screen, but some of these nodes are a little bit bigger than the others. This means that these bigger nodes are somehow optimized by the algorithm so that it makes it easier for the users to choose the root node. So for example, this centroid 39 is a little bit bigger than the others. Then it's going to yield a pseudotime graph. So this pseudotime graph will also show you how many cell states the trajectory predicted. So right now, it's predicting the trajectory to go from the top here down towards the bottom. And you can see that indicated by the graph down here as well. You have this different cell states over pseudotime on the x-axis. Okay, and so this may take a little bit to run, so I have some results ready in the next tab here. Okay, so it's going to yield a panel of genes over here. And if they are statistically significant, that means these genes are contributing or may be contributing to the transition from our mucus cells to our stem cells. So for example, I'll click on a certain gene, this CYCS gene, and you're going to be able to see the expression distribution of that gene. And it's slightly going up over pseudotime, meaning the CYCS gene is more expressed in our cancer, or I'm sorry, in our metaplastic stem-like cells. So to summarize, I'll go back to our slides. Okay, here again, this is performing pseudotime analysis where I wanted to see the trajectory from our mucus cells to our metaplastic stem-like cells. And in the results page, here's where Liam can explain a little bit more.
Right, so thank you, Julie, for the demonstration. And here are the genes that can potentially explain how our mucus cells transform themselves into the cancer stem cells. And therefore, the idea now is to gather the most statistically significant genes of these sets. And we try to see in our population of only mucus cells, which of the subpopulation have higher expression of these genes, which is what we are going to do next. So we have picked out some of these genes, as you can see on the panel on the right. And now we subcluster our dataset to only the mucus cells. And here you can see there are some subpopulation that have higher expression of these genes, which is denoted by the red color, whereas we would have some of them having lower expression of these genes. And now we really want to see in the red subpopulation which kind of biomarkers they have so that these biomarkers can be further investigated on how the mucus cells transform themselves into cancer stem cell. And actually, there are multiple methods on our platforms that can help us to extract these red cells cluster. And Julie will again demonstrate how you guys can do that.
Yeah. So let's call this set of 5 genes our stemness gene set. I'm going to go move over back to bbrowser X Pro, and you can see here on the left-hand side, I have them queried already. Again, I want to select only my mucus cells to look at, so I have a mucus subcluster saved here. This is going to be composed of our neck-like mucus cells and our pit mucus cells again. And if I click query, you're going to be able to see a higher expression of these genes towards the top of this cluster. So one way in which you can quickly select for this is by lassoing, meaning you can simply draw on the UMAP and then lasso this top area to select for these genes and then perform further analysis on them. Or you can reset that. Let's query this again. You can, down here at the bottom, select for how many genes are expressed. So this pale yellow bar here represents that 1 out of these 5 genes are being expressed in these cells, whereas this very dark red bar represents that all 5 out of 5 genes are expressed in a cell. So if I were to choose this range here, this means I'm selecting for cells that express 3 or more genes within my gene set. And then I can click subcluster to perform some further analysis. All right, so let's move back to our slides to talk about the results or the analysis we did after we had selected this. So the method of choice that we chose for this analysis was the lasso, just to get a quick look at what kind of other genes are expressed within this subpopulation of mucus cells.
Right. I believe that is a very convenient way and quick way of getting the subpopulation of interest. And again, we clicked on the biomarker function on the BB browser platform, I believe. And again, we have a list of genes that are differentially expressed in these clusters. And now, once the users get to this step, they can perform literature review to see which of these genes can potentially explain how mucus cells transform themselves into cancer stemness. So of these genes, we have the LY6E down at the bottom, which is one very nice example. Previously, there has been a study that show that these genes is seen in the cancer stem cells of multiple types of cancer. So yes, I believe we can even extract further insights on these genes. So not only is subclustering mucus cells and cancer stem cell a way to understand how they transform into one another, but we may also want to understand or see if there's any communication between these two cell types, which can be done by cell-cell communication. And Julie will now demonstrate how you guys can do that.
Yeah. So on bBrowser X Pro, we have the cell-cell communication feature over here on the left-hand side. So if we click that, all you have to do is enter the cell type metadata, which we have here, and you can also change the number of permutations as well. And once you click run, let's view what the results page looks like. So immediately you're going to see a heat map view of your sender cells on the y-axis and your receiver cells on the x-axis. So these are all the cells that are communicating with each other. And if you hover over a specific node, you'd be able to see the number of significant interactions, as well as what specific sender cell and receiver cell are playing a role in that interaction. What's interesting here is that down here on our sender cells, our pit mucus cells, we have a high number of significant interactions with metaplastic stem-like cells. And if we click open this node, we'd be able to see the interaction pair or the ligand and receptor pair that's involved in this communication, as well as the pathway that this is involved with and some statistics as well. So here, this MIF pathway, for example, is the macrophage migration inhibitory factor pathway, which is implicated in a tumor microenvironment, whereas these MK pathways have also been implicated in other types of cancers. So high expression of this MK pathway is typically associated with some other types of cancers. And we not only see that in our pit mucus cells, we also see a high number of significant interactions in our neck-like mucus cells here as well, interacting with the metaplastic stem-like cells. Okay, and then I also want to go over some other features in the cell-cell communication. So you're able to filter for certain pathways here as well. So if I only want to see pathways associated with the MIF, and see what sender and receiver cells I can filter for it that way. And then I can also switch over to this network view where I can look at the heat map, but in like a network. And if I hover over a specific cell type, you'd be able to see the interaction between that if you hover over the arrow itself. Okay, so with that said, let's switch back to our slides and take a look again at the results page.
Right. Can you move on to the next slide?
Yes.
Right. So until now, our finding and study is heavily based on the theory that the mucus cells have transformed themselves into cancer stem cell. This is a spoiler for next week content, but it also validates what we have found. So essentially, we have inspected some, so we inspected some spatial data set on a very similar type of tumorigenesis, but now on colon cancer. So previously of the genes that I say belonging to the secretory cells, we have the MUC5AC gene, which is a mucus producing gene. So when we see this gene's expression in a spatial data set of colon cancer, we see that the cells that express this gene is located and correspond to the epithelial cell, which is represented by the purple color. And the thing is that this epithelial cell population is situated right next to, and if not even blend into, the neoplastic cell, which are cancer stem cell. And the mention-worthy thing is that only the mucus-producing cell or the epithelial cell express the MUC5AC gene, whereas our cancer stem cell does not. So this is in accordance with what we had found in the gastric cancer study. So in summary, next slide, please. Next slide, please. So in summary, we have seen a downregulation of the genes that encode for secretory products, and we believe the cells that release these substances have been altered, and we propose a hypothesis that the mucus cells in our gastric pits have been replaced by cancer stem cells. We then propose several pipelines that user can perform and get a list of candidate genes that can explain our hypothesis of how mucus cells transform themselves into cancer stem cells. So that is the summary for our talk today, and thank you for listening. I believe we have a survey, don't
Yes.
we?
So yes, our colleague here is going to send out a poll soon.
Yes.
So you can go ahead, take time to answer that survey questions. In the meantime, we do have one question in the Q&A. So one person asked, do we have this option in Talk to Data as well? So sorry, I missed this when you typed this, but if you're referring to the data set, yes, we have this data set in Talk to Data, this one that we were analyzing throughout this talk. So you can find the study here on Talk to Data through this study ID down here at the bottom left-hand corner. Okay. You meant the cell-cell communication option. So currently only the cell-cell communication feature is only available on bBrowser X Pro. Yes, but soon we may be migrating these Talk to Data studies over to the bBrowser X Pro platform. However, I can't really give you a timeline on that yet, but that is to come in the future. Thank you for sending in questions. We'll give it a couple more minutes for people to send in any more questions you have, and also take time to answer the poll as well, which just popped up. So we're interested in basically building out more of the features that we have. So for pseudotime, if any of you guys have already played around with pseudotime or trajectory analysis, we'd like to see what more you're interested in seeing in that analysis. Same with cell-cell communication and differential gene expression analysis as well. So we're planning to have some enhancements to these existing features, but I just want to know, which would be most important or which should be prioritized for you guys. Okay, we'll give it another minute so that people can finish answering the poll. Okay, otherwise, thank you everyone for joining this week's SCSE Club. Please stay tuned for next week where we're going to be exploring spatial X and using that colon cancer dataset to explore some of our findings that we found today. Okay, thank you everyone for joining. Bye. See you next week.